All right, let's dive into one of the greatest detective stories in all of science, the mystery of heredity. Literally thousands of years, we were all stumped by a question that seems so simple on the surface, but is just profoundly complex. Seriously, think about it for a second. Why do you have your mom's eyes, but maybe your dad's nose? Or perhaps you've got a combination that feels totally new. How in the world does life pass down its instructions from one generation to the next, and how does it manage to create such incredible variety at the same time? For centuries, this was just a complete black box. Before we had any real evidence, the theories people came up with they were pretty wild. One of the most popular was something called the pre-formation theory. The idea was that a tiny, fully formed little person, they called it a homunculus, was just curled up inside either the egg or the sperm. I know, it sounds totally bizarre to us now, and that's because modern genetics has shown us the truth is so much more elegant. Traits are passed down through a chemical code, a language written in our DNA. And that journey from a tiny imaginary person to the discovery of the gene, well, that's the story we're exploring today. So, for most of human history, figuring this out was one of science's greatest unsolved cases. People obviously knew that, like begets like. Dogs have puppies, cats have kittens, but the actual rules governing it all? A total enigma. To even start solving it, the whole field needed a name. And that name is genetics. It's the science that's dedicated to understanding both heredity, that's the passing of traits, and variation, which is the wonderful stuff that makes every single one of us unique. Now, to finally crack this code, science was going to need a detective with a very, very particular set of skills. And you know what? That detective worked in probably the most unlikely laboratory you can imagine, a monastery garden. So let's meet the investigator who would find the very first crucial clues to this entire mystery. This is Gregor Johann Mendel. He was an Austrian monk who just happened to have a deep passion for science and, crucially, for mathematics. While so many others had tried and failed to find a pattern in heredity, Mendel brought something totally new to the table for biology, a super rigorous quantitative approach. He didn't just observe, he counted, he categorized, he looked for mathematical patterns in the living world. And his weapon of choice for this investigation? The common garden pea. And let me tell you, this was an absolute stroke of genius. Why? Well, pea plants grow super fast, they produce a ton of seeds, which means a huge sample size, and he could control exactly which plants pollinated which. But most importantly, they had these very clear, distinct, contrasting traits. In this, right here, this is where his genius strategy really shines. Instead of getting bogged down in complex traits that exist on a spectrum like human height, Mendel focused on 7 simple either-or characteristics. A plant was either tall or it was dwarf. Its seeds were either round or they were wrinkled. There was no in-between. By simplifying the problem this way, he could finally see the underlying pattern that everybody else had completely missed. Okay, so with his simple traits identified, Mendel got to work. He started his experiments, and by following the clues from his thousands and thousands of pea plants, he was about to uncover the fundamental rules of genetics, rules we still teach today. First, let's get a couple of key terms down. Think of a gene, like a general recipe for a trait, okay? Say the recipe for flower color. An allele, then, is a specific flavor variation of that recipe. So you might have the purple flower allele or the white flower allele. Mendel just called these unit factors, and they were the first big clue he needed to solve the case. And this leads us to another really crucial distinction. The genotype is the actual genetic code an organism carries, the combination of alleles it has. The phenotype is the physical trait you can actually see with your own eyes. So to use an analogy, the genotype is the architect's blueprint, and the phenotype is the finished building. So his first experiment was beautifully simple but incredibly powerful. Mendel took a true-breeding tall plant, one that only ever produced tall offspring, and he crossed it with a true-breeding dwarf plant. The result? Every single one of their offspring, the first generation, was tall. The dwarf trait had vanished. But Mendel had a hunch it was just hiding. So he took those new tall plants and let them self-pollinate. And in the very next generation, bam, the dwarf trait reappeared. Now, here's where it gets really cool. He meticulously counted thousands of these plants, and he found a stunningly consistent pattern. For every one dwarf plant, there were 3 tall plants, a 3-to-1 ratio. This was the smoking gun. I mean, think about what this meant. For the first time ever, A biological process could be predicted with a mathematical law. It wasn't just some random blending. There was a principle at work here. And this observation led him to his first major breakthrough, the law of segregation. He figured out that even though a parent plant has two alleles for a trait, it only passes one of them on to its offspring. These alleles have to separate or segregate when the sex cells, the pollen and ovules, are formed. This one simple rule perfectly explained that 3-to-1 ratio he kept seeing. But Mendel, being a great scientist, didn't stop there. He decided to up the complexity. What happens if you track two different traits at the exact same time? This is what we call a dihybrid cross. So he crossed a plant that had round, yellow seeds with one that had a wrinkled, green seeds. The big question was, are these traits linked together, or are they inherited totally independently of each other? And once again, the math gave him the answer. In that second generation, he found this beautiful, clear, unmistakable ratio, 9-to-3 to 3-to-1, for the different trait combinations. This elegant pattern proved, beyond a doubt, that the trait for seed shape was inherited completely independently of the trait for seed color. They weren't tied together at all. So there you have it. From these incredibly simple and patient experiments, Mendel deduced the 3 foundational laws of inheritance: the law of dominance, which explains why one allele can mask another; the law of segregation, which explains how those alleles separate into different sex cells; and the law of independent assortment, which explains that genes for different traits are passed on independently. He had done it. He'd cracked the code. But, like any good detective story, there are always some plot twists. And as science moved forward after Mendel, we discovered that genetics is even more complex and, frankly, more fascinating than he could have ever imagined. So let's look at a couple of cases where the rules seem to bend a little. First up, we have something called incomplete dominance. Now, in Mendel's peas, the tall allele completely dominated the dwarf one. End of story. But in some plants, like these beautiful snapdragons, neither allele is completely dominant. So if you cross a red-flowered plant with a white-flowered one, you don't get red offspring. You get pink. The heterozygote ends up being this perfect intermediate blend of the two. And then there's co-dominance, which is a little different from blending. A perfect example of this is the human ABO blood group system. If you inherit the A allele from one parent and the B allele from your other parent, you don't get some kind of blend. Instead, your blood cells express both traits fully and separately. You get AB blood type. The alleles are coexisting, or you could say they are co-dominant. Now, these exceptions don't disprove Mendel's work at all. In fact, they enrich it. They show how his foundational principles serve as the blueprint for a much larger, more intricate, and more beautiful structure. So let's talk about how his forgotten paper became the absolute cornerstone of modern biology. And this might be the most incredible part of the whole story: the unbelievable delay in recognition. Mendel published his groundbreaking findings way back in 1866, and the scientific community completely ignored him. It just sat there. He died in 1884, having no idea of the revolution he had just started. It wasn't until the year 1900 that 3 separate scientists, all working independently, rediscovered his paper and realized its profound importance. And what a revolution it was! Mendel's work laid the essential groundwork for literally everything we know about genetics today. It is the foundation for understanding how we inherit traits, how genetic diseases are passed on through families, and how we can use this incredible knowledge in medicine, in agriculture, in everything. So, I'll leave you with this final thought to chew on. Mendel's laws brilliantly explain traits that are determined by a single gene. But what about really complex traits like human height or skin color or intelligence? These don't come in simple either-or categories, right? They exist on this continuous, nearly infinite spectrum. This is the fascinating world of polygenic inheritance, where multiple genes all work together. And it just shows how far we've come, building upon Mendel's incredible blueprint. It's a powerful reminder that even after cracking that first fundamental code, the magnificent mystery of heredity just keeps on unfolding.